Okay.
Yeah. Next.
Good afternoon, everyone, and thank you so much for joining us for this week's Armor Seminar presentation. Before we begin, I would like to acknowledge that the Field Museum resides on the ancestral homelands of the Three Fires Confederacy, the Ojibwe, Odawa, and Potawatomi. It is with pleasure that I announce this week's speaker, Dr. Jessie Atterholt. To her academic background, Jessie got her BA from the University of Pennsylvania in 2009, working with the very famous paleontologist Peter Dodson. And then she went on to do her PhD at the University of California, Berkeley in 2015. Jessie is currently a professor at the Western University of Health Sciences in California, and she is an expert in anatomy from the skeleton to the bone and other tissues. And although she primarily uses this expertise to understand birds and their extinct relatives, as well as sauropod dinosaurs. And it is an special pleasure to introduce Jessie because we're actually very old friends. We met when she was doing her undergrad with Peter Dodson, and I was a PhD student at the time. So I think we met in 2008 in China, where we were working on some early Cretaceous birds, and our collaborations continue to this day. So please put your hands together for Dr. Jessie Atterholt. Thank you.
Thanks so much, Jingmei, and thanks very much to those of you who came out today and anyone who is on Zoom. Hello. I'm super excited to talk to you today about some obscure but very fun anatomy that I have been studying in birds and as applies to some extinct animals as well. I want to start by giving a big shout out to my collaborator on the projects that I'll be telling you about today. This is Dr. Matt Wadel. He is a colleague of mine at Western University of Health Sciences in Pomona, California, where we both work. Matt really specializes in sauropod dinosaurs. He really knows a tremendous amount about pneumaticity, but he's a general comparative anatomist as well. My own background has been a lot on birds, but I don't only study birds, so it's been a very fruitful collaboration for the two of us. So we have, at this point in time, developed a small research program that revolves around various questions that relate to strange anatomy inside of the vertebral canal, and I'll be telling you about two of these projects today. So just a little background context to be totally clear. When I'm talking about the vertebral canal, this is what I mean, the space that we find in a vertebra that's occupied by the spinal cord in life, and sometimes other soft tissues. And then also the other kind of jargon term in my title was archosaurs. So this is a basic phylogeny showing you the group of archosaurs. So this includes crocodiliforms, so crocodiles and alligators, extinct groups like pterosaurs, extinct dinosaurs, and then of course birds or extinct dinosaurs. Sorry, extant dinosaurs. So they're all considered to be archosaurs, but as paleontologists, to understand things that are extinct, we really care about using animals that are still alive today. And to do that, it's helpful to have this concept of an extant phylogenetic bracket, where we can look at the closest living relatives of those extinct animals. So in the case of extinct archosaurs, like pterosaurs and dinosaurs, the extant phylogenetic bracket is crocodiles and alligators on one hand and birds on the other. And Matt and I just happen to have kind of gone down a bird rabbit hole with our own studies. So that is where we focus on that side of the bracket. So the first project that I'm going to be telling you about today relates to bizarre respiratory structures that are found inside the vertebral canal in extant birds. So here is a CT scan through the neck of an adult ostrich, and you can see the neck vertebra, so the bone has high contrast, it's nice and bright white. Then inside of the vertebral canal, there's some soft tissue, which is really mostly the spinal cord, and that's the gray blob area. And then we also are seeing some black inside the vertebral canal on top of the spinal cord, and that is air spaces that are contained within parts of the respiratory system. So I think first it's helpful to start off with a little introduction to the avian respiratory system for anyone who's unfamiliar with it, because it's quite odd and very different from what we find in mammals and what we know in ourselves. So they do have lungs and trachea, just like mammals do, but the lungs are small and rigid. They don't get bigger and smaller as the bird breathes in and out. And they are connected to an elaborate system of what are called air sacs and diverticula, so just little divisions of the air sacs that spread throughout the entire body. So gas exchange still only takes place in the lung tissue, not in the air sacs. The air sacs are very thin membranous structures, but they're helping to circulate the air across the lungs in a very efficient way that essentially allows the bird to be uptaking oxygen when they breathe in and when they breathe out. And that's great if you're going to be engaging in a highly aerobic activity like flying. So these air sacs they can be categorized in general groups. We have cervical and thoracic air sacs and abdominal air sacs, and they are found between skin and muscle. They're found between muscle and muscle, and sometimes they even go into bone. And that helps to make the bird's skeletons very light and also very strong at the same time. So birds are full of lots and lots of air. So this is a CT scan cross-section of a baby ostrich. You can see one of the vertebrae, a dorsal vertebra up there at the top for context. And all the black areas are air. So tons of air in this animal. This is the case for lots of birds. This one was intubated prior to CT scanning it, and we filled the respiratory system with air, so you can really see everything that's going on. So lots of air inside these bizarre creatures. But what we really are excited about as paleontologists is being able to find osteological correlates of these soft tissue structures with the skeleton. Because if we can find marks on the skeleton that they leave behind, then we're able to make inferences about the presence of these soft tissues in extinct animals. And in the case of the air sac system, it's been very well documented that in entering bone or in relationship to bones, these air sacs can erode away the bone to leave behind pneumatic fossae, so concavities and pits in the bone, and also pneumatic foramina via which they actually enter into the bone. And so here are some examples of what these pneumatic fossae and foramina look like in extant birds. And then it's also been very well documented that we find very similar structures in extinct archosaurs. So pneumatic structures have been discovered in sauropod dinosaurs, long-necked dinosaurs, in theropods, the scary meat-eating ones, and even in pterosaurs as well. So a lot of the extinct archosaurs that I alluded to at the beginning of my talk. So if we come back to thinking about this in terms of the phylogenetic tree, it means that we have widespread evidence of an avian-like respiratory system in several of these extinct groups. Although I will note parenthetically that this doesn't apply to all dinosaurs, just some of the extinct dinosaurs like sauropods. So now back to these weird parts of the air sacs and diverticula that are actually entering the vertebral canal and sitting in contact with the spinal cord. For context, this is a diagram I'm going to be using a lot during my talk, so I just wanted to take a moment to explain it so that everyone understands what they're looking at here. So this is a sketch of the cervical vertebra of an adult ostrich in anterior view. You can see the vertebral body or centrum, and then above it, the vertebral arch, the two main components of the vertebra. The big hole in the middle is the vertebral canal. That's where I look a lot, and the spinal cord within it. And then because this is a cervical vertebra, there's also two lateral holes, and these are the transverse foramina for passage of the vertebral arteries as they make their way up the neck and into the skull to be part of the blood supply to the brain. And this is what those same structures look like in a dissection. And I'm sorry if I should have perhaps given forewarning that I was going to show a slightly gross picture. Well, some of you are using them. My apologies there. I dissect a lot of things, and I'm kind of well over the gross out factor by now. I forget. So this is really cool because we're seeing the same structures in a dissection. So this is a cross section through an ostrich neck. And we can see the structures that I've now alluded to several times, these weird respiratory system things inside the vertebral canal. They are called paramedullary airways. The more formal name is paramedullary diverticula. Paramedullary airways is easier to say. I'm just going to say PMAs a lot throughout the talk. So these paramedullary airways are diverticula that branch from the lungs and from the air sacs. They are found inside of the vertebral canal and also at intervertebral joints. And they're sitting in contact with the spinal cord. Extremely, extremely weird. why are they there? What are they doing? For reference, this is a color scheme that I'm going to use throughout the next few slides to make it easy to see these structures. So I'll use this sort of green color to highlight where we're seeing paramedullary airways and yellow for the spinal cord. And then also, these are a couple other related structures that will come up. These oftentimes are giving rise to and connecting with the anastomosis with these paramedullary diverticula. So there are supravertebral diverticula that sit on top of the vertebrae on the dorsal surface, as well as intertransverse diverticula that pass through those transverse foramina. So in birds, the vertebral artery is not the only structure that is passing through there. There's also diverticula, which is cool and interesting. So what's great is that while we can see these structures in a dissection, it's also very easy to identify them from a CT scan because they're filled with air, so they just appear as black space inside of the vertebral canal when they are present. And I, for my dissertation, happen to have a whole bunch of CT scans of dead birds. So Matt and I were able to conduct a survey for these structures among extant birds. Prior to our research, there were actually only 4 publications we were able to find that make any mention at all of paramedullary diverticula. And as you can see, 2 of these are quite old. They date back to 1908 and 1953. And these 2 publications are the only 2 that describe them in any detail. So the other 2 later publications kind of just mentioned them in passing. So prior to our work, essentially that means that we had a detailed description of paramedullary diverticula only in 2 species, representative of 2 major groups. As an aside, this is a really old phylogenetic tree. The topology here is definitely not accurate anymore, but I'm just using this to demonstrate the paucity of data prior to our study, and then in a few moments what we were able to add to them. So according to these 2 studies, paramedullary diverticula would present as tubes that are dorsal to the spinal cord, either one tube or a pair of tubes. That was what we knew about them. And we knew that they were present in turkeys and in pigeons. So in 2022, Matt and I published our CT-based survey of these structures in extant birds. We were able to increase our sample size up to 28 species and sample from 18 major groups. And for most of these species, we were able to sample at least 2 to 3 individuals as well. So what we found is that these structures are very, very broadly present. Most of the taxa that we were able to look in did have them. So these are all that had them. We find them in everything from as tiny as a hummingbird to as big as an ostrich. The species that are in boxes are the ones that are representative of the same clade. So we were able to get multiple samples from several clades, which was really great. We did find that they were absent in some groups, though. So they appear to be absent in grebes and loons. We did not see them in our 2 pisciforms that we sampled, which was 2 woodpeckers, and also did not observe them in kestrels. So it kind of makes sense that they would be, it's not shocking that they would be absent in grebes and loons because these are pursuit diving birds. They spend a lot of time underwater and they have overall highly reduced skeletal pneumaticity, period. So not having paramedullary airways makes sense. At this point in time, we do not have a good hypothesis as to why they would be absent in woodpeckers and in the kestrel. Although I will say I would like to definitely increase the sample size for falconiformes and pisciformes as we move forward with this project, just to be more sure that these structures are absent in these clades. But for the moment, our data suggests that they are for reasons unknown. And also, even though it makes sense that grebes and loons don't have them because of the way that they forage, several other taxa in our data set forage in a similar way, cormorants and ures. And we found that they did have paramedullary diverticula. So there doesn't seem at this time, based on our kind of more broad study, to be a clear functional signal for what these structures are doing when they are present. So now I'm going to tell you a bit about the different anatomical variations of these structures that we observed. So in some cases, we did see them present as 2 tubes dorsal to the spinal cord, as had been previously reported in the literature. And that's what we're seeing in the neck of an ostrich here. But sometimes we found that a pair of tubes could actually be more lateral to the spinal cord rather than dorsal to it. You can see that in a cute little bush tip here. Sometimes the two tubes can be in fact ventral to the spinal cord, and this was completely unknown prior to our study. So that was really interesting to find. But it only gets more weird from here. So sometimes there's not two tubes, there's actually three tubes. We saw this as well in the ostrich. So there's a lot of intra-taxonomic variation and even intra-individual variation. We would see these different morphologies in the same individual. Sometimes there's four tubes. This is what we saw in a green parakeet. And sometimes there's not separate tubes. They just kind of merge into one blob that sits on top of the spinal cord. And sometimes we get a very extreme morphology where the paramedullary diverticula completely jacket the spinal cord. So here the spinal cord is essentially suspended in space in the vertebral canal, completely surrounded by this obscure tiny little structure of the respiratory system. So really, really weird and cool and interesting to us at least. And this kind of takes that to the next level, where we have in this Turco an example. So we're looking within the sacrum here. And in this case, the paramedullary diverticula not only are completely surrounding the spinal cord, they have also gone on to pneumatize the sacral vertebra to the extent that there is essentially no bone left inside of the vertebra. So it's just an outer shell of bone retaining the morphology of that sacral vertebra, and with just paramedullary airway inside, and then a spinal cord suspended from that. So we saw some really extreme and really weird variations of these diverticula. So they can actually become quite large and take up the entire space of the vertebral canal and even the bone that forms the vertebral canal. So in this case, there is no vertebral canal anymore. Just the bone of the canal got pneumatized away. So where we then wanted to try to understand what all of these differences in morphology and in size were potentially correlating with. What are our potential explanatory variables to understand what's going on? Body size seemed to be an intuitive thing to start with, and we did see the largest and most elaborate paramedullary diverticula in the two largest birds in our data set, the ostrich and the brown pelican. But aside from those extremely large outliers, there wasn't a super clear pattern correlating with body size. So here you can see paramedullary diverticula in the bush tips, which is really, really tiny, and then in a turkey vulture, which is pretty darn large. And relative to the size of the spinal cord and the vertebral canal, paramedullary diverticula are quite similar between the two taxa, despite those differences in body size. We then took a look at patterns of variation across different vertebral regions. So of the 23 species in our data set that did have paramedullary airways, they were present in the cervical part of the vertebral column in every single one of them. So if a bird has, essentially it seems that if a bird has paramedullary diverticula, they will be found in the cervical series. As we move to the dorsal series, we only found them in 14 of those 23 species, and they were absent in 9, so they're becoming more rare. And then in this sacrum, they were present in only 5 species out of 23. So increasingly rare still. And at that, we only found them in the posterior sacrals. So they were not present at all in the more anterior sacral vertebrae, possibly because of the lumbosacral expansion and other weird structures associated, and large structures associated with the spinal cord in that area. And then finally, you're probably starting to get the gist of the pattern here. It's unsurprising that by the time we get to our caudal vertebrae, we only identified paramedullary airways in 3 taxa. And here you can see, I use the example in the pelican because it's really cool. I think that even to the bitter end, when the spinal cord is a tiny, tiny, tiny little dot, there's still paramedullary airways completely encircling it. We also hypothesize that there's probably variation through development, and this is really a direction for future research that we have yet to really dig into, but we intend to. But we have ontogenetic data for a couple of taxa in the data set. So these are just some very preliminary results, but we did find that in the ostrich chick, The paramedullary airways are approximately 36% of the cross-sectional area of the vertebral canal, and that goes up to 51% of the adults. So a pretty big increase in cross-sectional area. And we see a very similar pattern in great horned owls, where they go from taking up about 33% of that cross-sectional area to 48%. So certainly it needs to be unpacked with a lot more data, but these preliminary results suggest, unsurprisingly, that they get bigger as the animal gets older, matures, and grows. So in summary, what do we know about these structures? We know that they occur with very, very strong phylogenetic signal. For all of the clades for which we were able to sample more than one species, all the species within that particular clade either had these structures or did not have these structures. It seems the question of whether they vary with body size seems equivocal, maybe only larger in really extreme cases of large body size. That also needs to be sussed out further. We see a very, very strong correlation with different vertebral regions. So always present in the cervicals, rarely present in the caudals. So they decrease in the frequency in which we observe them, and also just in size and how elaborate they are as we move from anterior to posterior through the body. So this kind of seems to be indicating that perhaps function is distributing or influencing the distribution of these structures. So it seems that parts of the vertebral column that have a lot more movement, the cervical region, the neck, have these structures, and where movement is a lot more restricted, the notarium, the synsacrum, where vertebrae are fused up together, they're present less. So perhaps they're present in the neck to kind of help cushion the spinal cord, so to speak, during movement that occurs with the neck. Maybe, again, this is an initial survey, and all of this needs to be explored with additional data. And then finally, preliminary results suggest that development or ontogeny definitely does explain also some of the variation that we saw. So that's cool and exciting, but we're still paleontologists at the end of the day. So we want to know, how can we apply this to the fossil record? Can this help us learn anything about extinct archosaurs that had these avian respiratory systems? And to understand that, we need to have osteological correlates. And we did find some really clear nice ones. So first of all, pneumatic foramina, they're found elsewhere on the skeleton, but if we find them specifically inside of the vertebral canal, they can only have been formed by these paramedullary diverticula specifically. So pneumatic foramina in the vertebral canal are a really great one. Here you're seeing an example in a dry skeletal preparation of an albatross vertebra. And then in a CT scan, you can see a really, really large pneumatic foramen in the floor of the canal, the pelican, and the vertebral body or the centrum is being pneumatized via that foramen. We also found two other correlates. One is that sometimes they leave behind this sort of weird sculpturing or texturing within the canal. And the other is canal geometry. So some birds that have very large paramedullary diverticula will end up with this almost figure 8 shaped geometry to the vertebral canal, where the spinal cord is sitting in the lower circular part of the 8 and the paramedullary diverticula are sitting in the upper space of the 8, so to speak. So we have our osteological correlates. Now, what do we see in the fossil record? Well, we are absolutely finding evidence of these structures in the fossil record. So here is one example from a sauropod dinosaur called Giraffatitan. This was a study published by some of our colleagues in 2006, and we just were able to go and look at the CT scan that they published, this lovely transverse section through a vertebra, and you can see extremely clearly a nice pneumatic foramen in the roof of the vertebral canal. Similarly, this is an example of that sort of weird texturing in another sauropod dinosaur, this is Alamosaurus. This individual was a juvenile, so the vertebral centrum and arch were not fused. So we're actually looking at the roof of the canal because this is the piece of the vertebra that was the arch was popped off, and we're looking at the bottom of it, so to speak. So we see really cool evidence of these structures here in that form. I've also been working with some colleagues on exploring these structures in the Mesozoic bird Ichthyornis. And here we see a really nice example of that sort of figure 8 shaped geometry of the vertebral canal. I will also add, we're finding examples of pneumatic foraminae in the vertebral canal in this taxon too. And so we have so far sauropod dinosaurs, extinct birds, and finally now we can add to our list pterosaurs as well. So here is an example of a pteranodon where there's 4 consecutive dorsal vertebrae that all have pneumatic foramina in the floor of the canal in this case. And this is an extra interesting example because in our modern bird data set, where we saw this pneumatization in the floor of the canal, that always occurred with the complete jacketing of the spinal cord. So it kind of is teasing that maybe something as extremely weird was going on in the pterosaur, although of course we can't know for sure. So it's really exciting. This is only the tip of the iceberg in starting to find evidence of these structures in extinct animals. And so now I just like to share this information with as many colleagues as I can and tell people to start prepping out and looking in vertebral canals as much as possible. And that will be the same theme ultimately for the next project that I'm going to tell you about. So now we're going to shift gears and put an emphasis on sauropod dinosaurs rather than birds. So our second project deals with some very small, obscure, and hard to notice structures that are found in the caudal vertebrae of sauropod dinosaurs. And we have been calling them vertebral canal ridges until we figured out what they were. So I'm going to share with you the story of how we figured out what they were. But first, a little bit more about exactly what they are. So this is an example of these structures in an Apatosaurus caudal vertebra. So you can see there are these tiny little projections sticking out within the space of the vertebral canal. This is an example from Astropocadia, also a caudal vertebra. We are only finding these in caudal vertebrae. We have looked in other vertebrae, so far only see them in caudals. One feature of these structures is that they, in the sauropods, tend to be elongate from front to back or anterior or posteriorly. Here's an example in a Camarasaurus. This one's nice because it's actually broken, which in this case is a good thing because it gives us a really good view of one of these neural canal ridges or vertebral canal ridges. They're very hard to photograph if the neural canal is intact. So you can see not only are they elongate, but that these ridges are not extending to the anterior and posterior ends of the canal. They don't run all the way through it. This is an example of these structures in a Diplodocus, and we're seeing them in a CT scan, or rather a 3D model made from a CT scan. So this shows us that the size and the position of the structure is a bit variable. Sometimes they're appearing in the middle of the wall, sometimes on the very dorsal surface, sometimes on the very ventral surface. So we found them in all of those taxa by surveying museum collections. And we were able to collect additional data from previously published work. So we found that they occur in Rapetosaurus, Brontosaurus, and Europasaurus. And I'm excited actually to be here, and I'm going to go see the Rapetosaurus in person. So what we know so far about the phylogenetic distribution of these structures is that we have found them in every single sauropodomorph where we have been able to look for them. So the green check marks here mean they are present in these plates, and the question marks mean we don't know if they have them or not, and that's simply because we haven't at this time been able to find examples representing those plates with neural canals, or it has been CT scanned to the point where we can see these structures. So they appear to be extremely widespread. So the question is, what the heck are they? Well, our diagnosis of how they are appearing to us is that they are bony projections into the vertebral canal. They're located on the side wall of the canal. They're elongate from front to back, but they don't extend all the way to the ends. And their size and position might vary. It seems to vary serially and taxonomically. So we considered a variety of options to figure out what these things represent. Our first hypothesis was that they might be remnants of fusion of the neurocentral joint. So the way that vertebrae is developed is by fusing together two major pieces, the centrum or the body, and then the vertebral arch. So it's called the neurocentral joint. So perhaps these are representing just like a little bit of a leftover from where those two pieces of bone fused together. And we found evidence that suggests this is not the case. So we find these bony ridges in immature individuals where the neurocentral joint has not been fused. And you see an example here, again, in Rhepitosaurus, where the arch and the centrum are totally separate pieces, and also in an immature Alamosaurus. This one, the centrum is not preserved. We drew in a nice little circle to represent where it would be. So they're found in juveniles, and furthermore, they appear to be located dorsal to where the neurocentral joint eventually does fuse up. So not representing the neurocentral joint. We also considered the possibility that they might be associated with a soft tissue structure called the ligamentum flavum. A little fun anatomy background context. So this is a structure that connects consecutive vertebrae via a part of the vertebra called the lamina. So the lamina is essentially the part that's forming the roof of the vertebral canal, or the top part of the vertebral arch. This is a diagram of what the ligamentum flavum looks like to give you a sense of how it's connecting those consecutive vertebrae. And it does extend a bit into the vertebral canal. However, we ultimately ruled this out because even though it extends into the canal to make its attachment point, it's still only occurring at the very ends, or the dorsal roof of the canal. It doesn't extend into the lateral portions of the canal. And furthermore, upon further investigation, we found out that apparently ligamentum flavum is a structure that's specific to mammals and is not found in other vertebrate groups. So unlikely it would be that. We had also noticed from peering into the neural canals of our phylogenetic bracket representatives of extinct archosaurs that oftentimes there was an interesting geometry or shape to the vertebral canal. So we saw, this is an example in alligators and an example in an albatross, where we see something that actually looks quite similar to what we're looking at in sauropods. Looks like ridges into the vertebral canal, and it is. These ridges are separating soft tissue structures that are located there. So in the case of alligator, they're separating a really large dorsal spinal vein from the spinal cord. And as we already know, in the case of the bird, they're separating a spinal cord from the supramedullary diverticula. However, these are distinctly different from what we're seeing in the sauropods, because here the ridges extend all the way to the ends of the canal. They definitely weren't doing that in the caudals of the sauropods. And furthermore, they change the entire geometry of the canal. So you can take a cross section at any point and it's going to have this kind of figurate appearance. And that was not quite what we were seeing in the sauropods. So our next hypothesis was that they might be bony spinal cord supports. And in order to explain fully what exactly that means, I'm going to go off on a fun little general anatomy tangent. So the central nervous system, the brain, and the spinal cord are protected by three specialized layers of tissue called the meninges. The outermost one is the dura mater. It means the tough mother. So this is a relatively thick, very protective layer. The next one is called the arachnoid mater, and this borders the subarachnoid space, which is filled with cerebrospinal fluid. The arachnoid mater gives off these wispy, delicate little projections that dangle down into that space, and they look kind of like cobwebs. So it's the spiderweb mother. And then finally, the pia mater. Oh, before I get to the pia mater, I forgot. I included a nice picture of the dura mater and the arachnoid mater. So this is from a dissection from one of our donors in our lab at Western U. So you can see the dura mater being pulled to the side there. It's quite thick, it's held with the forceps, and you can see the delicate spiderweb-like projections of the arachnoid mater. And then the pia mater, it means the soft or the gentle mother. It's very, very thin layer, and it's kind of just attached directly to the outside of the spinal cord and to the brain. But the pia mater also forms another structure that we call denticulate ligaments. Denticulate ligaments are projections, lateral projections of pia mater that anchor the spinal cord to the dura mater. And they occur segmentally in between where spinal nerves are emerging from the spinal cord. So this is a picture of what they look like. Again, this is in a person, in a human. So the dura mater has been reflected. You're looking down onto the dorsal surface of the spinal cord. The black or the dark squiggly things are spinal veins. Pia mater, it's essentially kind of transparent and invisible, but technically it's the layer that's directly on top of the spinal cord. And then you can see denticulate ligaments, those kind of gentle triangular shaped things that are connecting, going from the spinal cord to the dura mater. So those are our projections of pia mater that support the spinal cord. So that's what they are. And now another important background factoid that we have to bear in mind as we explore what these might be in sauropod dinosaurs is that in mammals, the epidural space, so exists a space between the bone and the periosteum of the vertebral canal and the dura mater, and it is usually filled with fat. In non-mammalian vertebrates, there isn't an epidural space unless you have a soft tissue structure insert itself between the dura mater and the bone of the periosteum, or the bone of the vertebral canal, as is the case with paramedullary diverticula, but that is an aside. So generally speaking, the dura mater is just directly fused to the bone of the vertebral canal. We did a literature search for evidence of bony spinal cord supports. So these would be bony projections into the vertebral canal that those denticulate ligaments are anchoring to. And we found a paper that reported on finding bony spinal cord supports for anchoring the denticulate ligaments in little embryonic salamanders. So this is an example there, and this dates back to 1973 when these were first being discussed. We found another paper that surveyed these structures broadly across vertebrates. They found them to be very common in salamanders. So here's two other examples, including in giant salamanders. I always love those guys. They kind of creep me out, but they're really cool. So this is not only interesting because they're broadly present in salamanders, but we're also seeing that they vary serially. In salamanders, they're actually present throughout almost the entire vertebral column, but they are appearing slightly differently in different regions of the vertebral column. They also reported finding these structures in salmon and in trout. That's what we're seeing here. So this was really exciting. Oh, here's an example that Matt and I found in a tuna in a museum. It's exciting because we finally had a structure that met our criteria. So these are bony projections into the vertebral canal. They're located on the side of the canal or the lateral wall. They don't extend all the way to the anterior and posterior ends, and they're variable in size and position across taxa and serially in different vertebral regions, or even serially in the same vertebral region. So we think this is strongly suggestive that these structures are indeed bony spinal cord supports in the caudal vertebrae of sauropod dinosaurs for anchoring denticulate ligaments. We hypothesize that these soft tissue structures would look something like this, with the dura mater fused to the bone of the vertebral wall or vertebral canal, and the denticulate ligaments attaching to those ridges and suspending and anchoring the spinal cord. So the previous paper that I had alluded to, Sketches and Believe It, who surveyed a lot of extant amniotes or extant vertebrates, they reported that these structures are only present in bony fish, lungfish, and salamanders, and absent everywhere else. So our results show that we have, for the first time, evidence of bony spinal cord supports in an amniote, in at least an extinct dinosaur. So that was really cool and interesting and rather unexpected. And we think this is suggestive that perhaps more data needs to be collected on other amniotes, especially turtles and snakes. Sketches and Believe It did check them, but I think only one example of each. And they even found in an early lizard embryo, they found remnants of, or they found bony spinal cord supports that actually disappeared later in ontogeny. So it's a weird characteristic that we don't know enough about yet in amniotes, I would say. But we have evidence of them in dinosaurs, so that's really exciting. The next question, of course, is why are they there? Why are we only finding them in the tail? So we are thinking that perhaps it has something to do with stabilizing the spinal cord during lateral movement. A lot of the extant groups in which we find bony spinal cords today, bony spinal cord supports today, have a lot of bilateral movement when they locomote, salamanders, fish. And then why specifically in the tail of sauropods? Well, it's been well documented now that a lot of sauropods use their tails like weapons. Some of them have spikes on their tails or clubs. Some of them we're probably using them as just giant whips. So they would be whacking them around a lot. There would be high impact on the tail. It makes sense that the spinal cord might need a little bit of extra support. So that all sounds really great, right? But we don't care just about sauropods. We really look in every vertebral canal that we can. Like, we're ridiculous and silly. Every time we walk in a museum, we're looking at all the exhibits, or we're in collections and looking. It's really kind of sad how often vertebral canals don't get checked out. Anyway, we look everywhere we can, we try to collect as much data as we can. And this is one example of a place where we found these structures that is a non-sauropod. So this is in a hadrosaur dinosaur, and these are dinosaurs that actually have their tails very stiffened with ossified tendons, so they weren't undergoing, presumably, although if anyone knows more about biomechanics of hadrosaur tails than I do, which is extremely little, correct me, but presumably not undergoing a lot of whipping the tail around and lateral motion. So that seems a point not in our favor, trying to explain what these are for, but that's where we currently stand with the project. We're continuing to collect data. I wasn't able to include the photo in this, but we have found these structures in caudal vertebrae of Allosaurus, so sauropod dinosaur as well, or sorry, a theropod dinosaur as well. So just continuing to learn more about who has them. So in summary, we're finding that you can find evidence of really cool, interesting structures and interesting soft tissue, evidence of interesting soft tissue anatomy by looking in the spinal or the vertebral canal and exploring what's going on. And we encourage other people to go out in museums and collections and in displays, start peering into canals and looking for bony spinal cord supports, as well as pneumatic foramina and evidence of paramedullary diverticula. And with that, I thank you very much for your attention, and I'm happy to take any questions that you might have.
Oh, first of all, what's that?
Oh, it's just, this is totally tangential. It's just a pretty picture that made me happy.
Remember.
Oh, I see the label now at the bottom. I'm like, oh, okay, wait a minute, sorry.
Yeah, it's a whale cervical series. We were camping, my husband and I were camping at Pismo Beach last summer, and outside their visitor center, they had this, and I just thought it was so pretty. It had the wildflowers growing around it, so.
Cool. So yeah, so my question was, you saw all this variation in the paramedullary airways,
Yep.
even within a taxon, which I thought was weird. So,
Yep.
but they were all specimens that were dead when you CT scanned them. So do you think that, like, I mean, I guess, is there a possibility that this variation is the result of like things getting gross, like after death, like kind of maybe affecting shit?
I think a little of it might be. I think that, so I mentioned that one of the morphologies we see is like just a blob on top of the spinal cord instead of separate tubes. And in that case, it might be just the resolution of the CT scan wasn't high enough to pick up on like the membranous walls separating different tubes, or maybe it degraded and they blobbed together. But I think it's only having a minor effect on our data. And what's actually really interesting, I didn't mention this, trying to minimize the details to make the talk efficient, but some of the data that we looked at are data I collected together with Emma Schachner, who does a lot of really beautiful studies on the respiratory system of modern birds. And for her research, Emma really wanted to intubate the birds and inflate the respiratory systems. So for some of the birds, that was done. But for some of the birds, that wasn't done. And we're still able to see the structures even when they have been intubated and have their sacs. So, yeah. Yes.
So you talked a little bit about when the paramedullary diverticula fills the synsacrum.
Yes.
So I have two questions related to that.
Yes.
The first being, is there any sense that you have about biomechanically what that does to the synsacrum and its association with pelvis? And secondly, do you have any sense of how that interacts with the lumbosacral organ?
That is a really good question. So in answer to the first, I don't have no guesses or knowledge about how it might affect things biomechanically. I would assume it would make the bone pretty weak if it's like has no internal structure anymore and it's literally just an outer shell of bone, but the animal seemed to be doing fine. So, I mean, it died eventually, but it was like, it didn't die from like collapse of the sacral region and smoking of the spinal cord. And then when it comes to lumbosacral organs, this is one of the reasons I think we don't see paramedullary diverticula in that part of the sacrum.
In the anterior part?
Yes,
Okay.
exactly. We're usually finding them if they aren't there anymore.
Okay,
Yeah.
very interesting.
Yeah, I know, right? It's so weird. So lumbosacral organs are another thing that we're really interested in because again, weird stuff in the vertebral canal. Yes.
I have two questions.
Yes.
So question number one is woodpeckers and falcons, the two that you inexplicably found.
Yes.
Though it strikes me, those are two birds that are known for hitting things a lot very fast. And I was wondering if it was possible that it was somehow related to that. I don't know why it would produce that effect. Like woodpeckers are always hammering their heads on things, and falcons are like diving at things at like hundreds of miles an hour.
Yeah, so my thought when I first saw that they didn't have them was, I was just like, this is extra confusing. You would think that like if these structures are present to kind of cushion the spinal cord, they would really, really need them in these taxa. But we actually had a really nice discussion of this paper in Jane May's lab group earlier this week, and I can't remember, Alex maybe, one of her students pointed out that maybe the lack of them is being as a form of protection in and of itself. Because the spinal cord can't really like bounce around as much if there isn't space for it to bounce around in, so to speak. So maybe that's what's going on. Like I said, I really want more data for those groups before I start going too much down the rabbit hole of trying to think of functionally why they would not have them. But yeah, that's the current best explanation that I.
But pelicans have it, right?
But pelicans have it,
Yeah.
yes. So it's like the exact opposite there. Yeah.
Yes, okay. Can I ask my second?
Oh, yeah, I'm so sorry.
So the second one is like, are there other like foramina you might find in the neural canal besides pneumatic foramina? Because I know that at least in some sort of odd fossils I've seen have built like a really big pit kind of on the dorsal surface of the centrum. And that's unrelated, or?
That is unrelated, so that's another really good question. So for anyone who is seriously interested in this, I'd be happy to send you papers and talk to you more about it afterwards. But that is a little word of warning. So there are neurovascular foramina within the vertebral canal as well. Especially, I've noticed the same in sauropods. They usually tend to be in the floor of the canal. So you have to be able to distinguish a foramen that's for vasculature from a foramen that was resulting from pneumaticity. And it's difficult and it is nuanced, but in short, the neurovascular or canals or holes for vasculature tend to be more rounded and regular looking because the bone formed around them, right? They developed first, then the bone formed. Versus pneumatic foramina, the bone developed and then the air sacs came along and started eroding away the bone. So they tend to have sharper edges and be a little bit more irregular in their appearance. And then the ideal is that you have a CT scan and you can follow that hole and see what it's doing once it gets inside. And is it connected with a giant space that it probably eroded? Then it's pneumatic. And is it not? Then probably it's vascular. Yeah. Great question. Yes.
I have like 3.5 or 2.5 questions. There were 3 birds I was really curious about for this. You might only be able to answer one of them. So the first one is probably the easiest one. I'm curious about mergansers with regard to these tubes in the spine. You're diving ducks.
Yeah.
Did you look at any ducks for this?
We had mallards.
Okay.
And they also, this is one where I actually only had chicks. I didn't have adults.
Okay.
So what we found in mallards is that they were present,
Okay.
but that's a mallard, which is a different, sorry, what you're asking about. So I think one of our next steps is to try to collect more data on birds that are doing more diving and spending more time underwater.
Okay, my follow-up question, my next one is the penguin. What do penguins do with this?
I don't know yet. I really want to know, but gotta get my hands on some penguin CT scans first.
The third group is like kind of a diverse group that's spread across like several taxa. And that's cursorial predators like Seriema, secretary birds.
Also don't do.
Okay.
So that is definitely, we have 2, I mean, there's so many other projects to come out of this. Our top priority is just starting to identify these structures in extinct animals. But another thing I really want to do is a more refined and expanded analysis where we are intentionally sampling from particular ecologies and biomechanical groups. And see, that way we can try to refine if there is any correlation between presence or absence and morphology of these structures with certain behaviors, et cetera.
In relation to what he said with hitting things, the Seriema is this unique thrash that would be fascinating to look at.
That would be so cool. I know. Yeah, thank you for the idea. Was that all?
That was a 2.5, yeah.
Okay, awesome.
I'm curious about the pelican having that fully sheathed spinal cord. Are there structures that help the spinal cord in place within the pneumatic diverticula, or is it literally just kind of bouncing around in there?
To my knowledge, there's not. So when the diverticula get into the spinal cord, or sorry, into the vertebral canal, they are pushing their way in between the bone, the bone and the dura mater. So they do still have denticulate ligaments anchoring to the dura mater, but the dura mater is now not fused to the bone of the vertebral canal. So those denticulate ligaments are also not extending to anchor themselves to bone. So as far as we're able to see at this time, and it'd be really cool to do a nice histological study and look at in greater detail the different tissues and what's going on. But from what we can tell as of now, nope, there's nothing. It's so weird, yeah. Any other questions?
You know about all the CT scan that's now, CT data that are now available on source for
I
birds?
do as of Monday
Oh,
from your
okay.
museum, right?
Yeah,
Yeah, that I had known prior, and then when we were discussing people in Digby's lab, they were like, so do you know about these scans? And I was like, that sounds amazing, so.
cool, yeah.
Yes, thank you so much, that is absolutely going to be a wealth of new data for this project.
Yeah.
I'm excited. Okay, well, thank you so very much for your attention, everyone, and for really clear